Welcome to the lecture series in food biotechnology. As we have been mentioning earlier, the combination of food, which is an essential part in every organism's life, combining it with the technological advances which will help us to produce more or to increase the shelf life or to process the goods more is an exciting avenue in the field of food science and nutrition. In this particular lecture, we are going to talk about the methods of plant transformation. Plants, as you know, are the primary producers of food. Whether you are the first consumer, you are a vegan or a vegetarian, or whether you are a non-vegetarian, the primary source of food of all is plants. And in the field of food technology, we have various methods by which we can improve the plants so as to get a nutritious composition or to increase the nutrition or sometimes to reduce the fertilizers and the manures which you add to increase the yield of the crops. So, when we talk about the methods of plant transformation, and as a science, if you are going to look into it, there are certain criteria which have to be followed when we are doing the transformation. What are the biological requirements for transformation? So, the basic thing what you require is the gene. In what aspect are you going to improve the plant concern? And if you know the gene, is this gene a part which is taken up from any plant, or is it from any other organism like an animal or a protozoan or a bacteria? The second aspect is the availability of the target tissue, including the cells which are competent for plant regeneration. Okay, so once you have isolated which is the gene or which you have pinpointed, which is the gene which you are going to introduce into the plant, the second question is, in the plant, where are you going to put it in? In which tissue? Is it the stem? Is it the leaf? Or is it the chloroplast? Which tissue are you going to transform? Then the major criteria here would be that whichever is the plant part which you choose, you should be able to regenerate the whole plant so that the whole plant will contain the gene which you have introduced into the plant tissue which you have concerned with. The third aspect is a method to introduce the DNA. So how are you going to introduce the DNA into the plant? That is the third aspect. And then would be the procedure to select and regenerate the transformed plants at a satisfiable frequency. So these are the biological requirements. So what are the practical requirements? Beyond the biological requirements to achieve transformation and the technical requirements for the verification of the reproducible transformation, the desired characteristics to be considered in evaluating alternating techniques or developing new ones for cultivar improvement include: 1. A high efficiency, economical and reproducible protocol which would readily produce many independent transformants for testing. Whichever is the method which you choose to introduce the gene into the tissue concerned, it should be economical, it should be reproducible, and also once you have transformed the tissue concerned, you should be able to get a large amount of progeny so that every time you don't have to go back and transform the tissue concerned. The second aspect is safety to the operators, whoever is doing the protocol, avoiding procedures or substances requiring cumbersome precautions. That is, whichever is the protocol you choose, it should be easy, it should not be causing damage either to the operator, or you should not be able to have so much of precautions to be taken that it is very difficult to do the procedure concerned. And also, carcinogenicity should be avoided. For example, potential carcinogenicity of the silicon carbide procedure. DNA incorporation using silicon carbide whiskers is a normal protocol for gene introduction, but it is highly carcinogenic. So normally we don't apply or use that particular protocol. Technical simplicity involving a minimum of demanding or inherently variable manipulations such as protoplast production and regeneration. Whichever is the protocol or the procedure you choose, it should be simple. And also, once you have transformed, you don't want to have much of mutations or variations happening to the tissue concerned, because in turn that can reduce the effectiveness of the gene which you have introduced into the tissue. Minimum time in tissue culture to reduce the associated cause and avoid undesirable somaclonal variation. If you have to grow the crop in the lab conditions for a huge amount of time, that requires time, effort, and it would also involve a huge amount of cost, so you don't want it. Then stable or uniform transformants for vegetatively propagated species, or fertile germline transformants for sexually propagated species. So depending upon whether your explant is vegetatively propagated or sexually propagated, you require stable and uniform transformants in both the cases. Simple integration patterns and low copy number of introduced genes to minimize the probability of undesired gene disruption at the insertion sites or multi-copy associated transgene silencing. When you have a complicated insertion procedure, instead of a straightforward one-time insertion, sometimes the genes might not get inserted in the site you want, or it might get eliminated, or it might get broken in between, thereby not giving you the product which you are looking for. Or in the other case, if the number of genes which you have got inserted is much more, what you call as a high copy number, then the tissue itself has what you call as a natural tendency to silence the genes concerned if it is not required. We call this as the transgene silencing. So that is also has to be avoided. Stable expression of the introduced genes in the pattern expected from the chosen gene control sequence. So you require the expression of the gene once it has been inserted. The last is the particle gun or biolistics. The methodology for utilizing particle gun, we already saw in the last lecture. As a recap, actually what we do here is, we have the gene gun barrel at this area. So you have the DNA, plastic coated DNA at this particular structure, gold particles. The DNA coated with gold particles are incorporated here. You apply vacuum here and you apply the pressure. So, and you have a wire mesh which will so when you apply vacuum and the pressure, like a bullet going from the gun or delivered from the gun, the DNA with the gold particles will penetrate and it will fall onto the tissue which you have kept here. Whereas the plastic which has been, the disc onto which the DNA has been coated, it will be stopped by the mesh which is present here. So this particular technology for plant transformation was developed by Sanford et al. At the Cornell University in 1985. And the steps for plant transformation include: 1. Isolate the gene of interest from the source of organism. Develop a functional transgenic construct including the gene of interest, the promoters to drive the expression, codon modification if needed to increase successful protein production, and marker genes to facilitate tracking of the introduced genes in the host plant. Irrespective of whichever is the protocol you use for developing a transgenic plant, these are all the basic steps which are required. That is, isolate the gene, develop a construct, have a suitable promoter, optimize the codon if it is required, and then introduce marker genes so as to facilitate or so as to identify the explants which have been transformed from the non-transformed explants, and you have to select them because you want to grow only the transformed explants. So these are the fundamental steps in any transgene technology. So incorporate into an useful plasmid, introduce the transgenes into the plant cells, and regenerate these transformed plant cells. And then you have to test. See, you say that you have introduced the gene, but you need to get a proof that yes, this transformed gene is inside the particular cell concerned. So you have to test the trait for performance or for gene expression at the lab level, at the greenhouse level, and at the field level. Particle bombardment also plays an important role in the transformation of organelles such as chloroplasts, which enables engineering of the organelle encoded herbicide or pesticide resistance in crop plants and to study the photosynthetic process. When you do a nuclear transformation, that is you introduce the transgene into the plant nucleus, there is a fear that it might get spread because of ovum or it might get spread via pollen, and so many other difficulties are there. So to avoid that, we have a methodology, what you call is chloroplast engineering. So as you know, you will have millions of chloroplasts within a cell, depending upon whether you are taking a leaf tissue or a stem tissue. So it's possible to transform these chloroplasts and then get those traits expressed in the chloroplast. So in the case of developing a plant for herbicide resistance or for pesticide resistance or for improving the photosynthetic efficiency, chloroplast is a better target than any other tissue concerned. Limitations to the particle bombardment method relative to agrobacterium-mediated transformation includes frequent integration of multiple copies of the transgene at a single insertion site, rearrangement of the inserted genes, and the incorporation of the transgene at multiple insertion sites. The other methodology which is normally used is for plant transformation is agrobacterium-mediated gene transfer. Agrobacterium tumefaciens is a rod-shaped gram-negative bacteria found in the soil, and it is the natural causative organism of what you call is a crown gall disease. The bacteria enters the plant through cuts or wounds which is present in the root or the shoot system or the stem. The bacterium then inserts its DNA and stimulates the plant to grow swollen galls or tumors. Agrobacterium tumefaciens is capable of inter-kingdom DNA transfer, thus making it a potential vector in the production of transgenic plants. See, normally agrobacterium is present in the rhizosphere. If there is a wound in the root system or in the stem which just borders the root system, from the root, the agrobacterium would move towards the wound site and it is capable of transferring its DNA into the plant system. So this is the first example of inter-kingdom transfer, as a result of which the plant will form small tumors or crown galls at the site where the wounding has happened. Transformation using this organism has its advantage in identification of the transgenic expression. The presence of the gall represents the manifestation of transfer. So if the plant starts developing a gall, it indicates that gene transfer has already happened and the expression of the additional DNA is present in the plant cells. The application of this method is found in both scientific methods as well as the commercial ventures. Let's see the procedure for plant transformation. So you have the plant from where you have taken the leaf. You have made, for example, so you have made small discs. You have cut small discs out of that. This leaf disc is transferred into a medium or a Petri plate where you are growing the genetically transformed agrobacterium, which contains the gene of your interest. So you incubate this leaf disc in this solution containing the genetically modified agrobacterium for 2 to 4 hours. then, or if for, you can even for 24 hours, then you can see the time depends upon which is the plant. Here we have taken leaf disc, so 24 hours is enough. But depending upon the plant and the explant concerned, this incubation time can vary. So then you have to transfer these incubated leaf discs into a plate containing normal medium, plant tissue culture medium. So some bacteria would have transformed or transferred their genetic material, including the gene of interest, into these leaf discs during this incubation period. So you allow those bacteria some time to start dividing, and also the plant cells some time to adapt to the infection. Then we actually put them in selection medium containing some antibiotics so that the extra bacteria can be killed. And also in this medium, only those particular cells which have got the DNA from the transferred agrobacterium will continue. So normally we would have coupled antibiotic resistance. So if you add the antibiotic to the medium, only those leaf discs which have got the transfer of the DNA containing the resistance gene would start multiplying here. So once it has started dividing and you get sufficient amount of callus, you transfer this to what you call as a shoot induction medium. Let it form a shoot. Once it has grown a little bit, grow it to a root induction medium, let it grow a root. So once the plantlet has grown and has grown a little bit, you again transfer it to the pot in the greenhouse. So this is the normal procedure of how you can use agrobacterium mediated transformation for growing a transgenic plant. So the preliminary step in all these is the capability of the plant cells to be totipotent. Totipotent meaning if any part of the plant you take, it can dedifferentiate to form a meristematic tissue, and that dedifferentiated meristematic tissue can again form a plant provided you give it the sufficient nutrients. Tissue culture is defined as the culture of plant seeds, organs, tissues, cells or protoplasts on nutrient media under sterile conditions. So these are the steps which are normally followed. So you have a nutrient medium here, you take your explant and incubate it, and then subsequently subculture this in the same medium for adding growth regulators for some time. These will start developing, we call this as the subculturing. These will start developing into calli, which is the dedifferentiated meristematic tissue. Then those can now form individual shoots. You can now separate out each of these shoots, put them in the rooting media so that they can start forming the roots. So these are the roots which are formed. And then you finally transfer them to the plant. So this is again a picture showing plants growing in vitro in the lab, in a tissue culture lab. So depending upon the explants which you have taken, it can be stem tip, root tip, pollen grain, part of the embryo, cotyledon, tip of the meristem, anything. Provided you give the proper nutrient medium, it can form a new plant. Tissue culture is widely used to produce clones of a plant, and the methodology is called as micropropagation. Different techniques in plant tissue culture may offer certain advantages over traditional methods of propagation. The advantages are: one, you can produce exact copies of plants that will produce a particularly good flower or a fruit. To quickly produce matured plants, to produce multiples of plants in the absence of seeds. So if there is no sexual method of reproduction and seeds are not formed, then the plants have to be vegetatively propagated. Tissue culture actually gives an in vitro scenario where the plants can be grown inside in a lab, and then you can mass transfer it to the greenhouse and culture. So that way you would be preserving the quality of the fruit or the crop or the yield you want without resorting to sexual reproduction. Then regeneration of whole plants from the cells which have been genetically modified, the production of plants in sterile containers that allows them to be moved with greater reduced chances of transmitting diseases, pests, and pathogens, the production of plants from seeds that otherwise have a very low chances of regerminating, for example, orchids. They propagate very slow, normally seeds are very less, so propagation becomes very difficult. To clean particular plants of viral and other infections and to quickly multiply these plants as a cleaned stock for horticulture and agriculture. Single cells, that is plant cells without cell walls, otherwise you call them as protoplasts. Leaves, portions of stems, portions of roots, all of these can be used as an explant for tissue culture, and provided you give the optimum media containing the required nutrients, all of these explants can grow up to give rise to saplings or seedlings. Preparation of plant tissue culture is performed under aseptic conditions under HEPA filtered air, which is provided in a laminar airflow cabinet. Thereafter, the tissue is grown in sterile containers such as petri dishes or flasks in a growth room with controlled temperature and light intensity. See, normally plants grow outside. Sunlight influences that. Temperature of the area also influences that. So when you grow in the lab, you have to adjust the temperature as well as the light intensity so that the plants are independent, they can photosynthesize, at the same time grow properly. Living plant materials from the environment are naturally contaminated on their surfaces with microbes. So the surfaces are sterilized using the chemical solutions, usually alcohol or sodium hypochlorite or calcium hypochlorite. The sterile explants are then usually placed on the surface of sterile solid culture medium, but sometimes they are placed directly into the sterile liquid medium. So the media can be either solid or liquid depending upon what is the material which you have taken. And normally if you require cell suspension cultures, we use liquid medium. Solid and liquid media are generally composed of the inorganic salts plus a few organic nutrients, vitamins, and plant hormones. And the solid media is prepared from the liquid media with the addition of a gelling agent, normally either phytogel or the purified agar. Composition of the medium, particularly the plant hormones and the nitrogen source, the nitrite versus the ammonium salts and the amino acids, have great effect on the morphology of the tissues that grow from the initial explant. For example, including the nitrates and ammonia, you can also add vitamins and growth regulators like auxins and cytokinins. So in this case, for example, if you add an excess of auxin, you will get more amount of roots formed in the explant. Whereas if you add excess of cytokinin, you will see more amount of shoots getting formed. And if you have a balance between the auxin and the cytokinin, normally you will get the meristematic tissue or the callus. So as we have been explaining, depending upon the composition of the medium, where you are going to use an extra auxin or a cytokinin, and if you are going to use a balanced ratio of auxin and cytokinin, the products which you get in tissue culture can be different, like whether you have a meristematic callus, you have more amount of rooting as you see in this picture, or you see the development of the shoots as you see here. As shoots emerge from the culture, they may be sliced off and rooted with auxin to produce plantlets, which when mature can be transferred to the potting soil for further growth in the greenhouse as plantlets and under control. So you see this is a meristematic callus and you see the shoots which are coming up. You can see the multiple shoots which are coming up here. Each of these multiple shoots can be cut, placed in a medium containing auxin, and then individually each of them would develop into a plantlet. The specific differences in the regeneration potential of different organs and explants have various explanations. The significant factors include differences in the stage of the cells in the cell cycle, the availability of or the ability to transport endogenous growth regulators, and the metabolic capability of the cells. So, the choice of the explant and also what is the part, like are you using a stem or a leaf or a root, each of them and the tissue concerned have different rates of what you call as a cell cycle. So even in the tissue culture, that it will influence the regenerating capacity, whether it is going to form a good amount of callus and subsequently you can get more amount of plantlets out of it. The most commonly used tissue explants are the meristematic ends of the plants, like the stem tip, the axillary bud tip, and the root tip. These tissues have high rates of cell division and either concentrate or produce the required growth regulating substances, including auxins and cytokinins. Shoot regeneration efficiency in tissue culture is usually a quantitative trait that often varies between plant species and within a plant species among subspecies, varieties, cultivars, and ecotypes. See, suppose you are taking rice itself, there are different cultivars of rice. We talk about IR8, IR10, Ponni, Basmati, whatever. So each one of them, if you put, suppose I say I'm going to take the endosperm and culture it, all would be totipotent, all would give, provided you put it in the proper nutritive medium, it will give rise to callus. But the amount of callus, the time taken for producing that amount of callus, and then subsequently forming a shoot and a root, each will vary in each of these species in subvarieties. So accordingly, each scientist or a person who is going for this would have to choose that particular variety which gives the maximum amount of reproducibility, that is totipotency, more amount of platelets, because ultimately if clonal propagation is your what you are looking for, you require more meristematic tissue which has more amount of callus and then more amount of shooting and rooting from those particular callus which you have chosen. Three common pathways of plant tissue culture regeneration are propagation from the pre-existing meristems, which is shoot culture or a nodal culture, organogenesis, and also non-zygotic embryogenesis. We will be dealing with the various protocols in the subsequent lectures. Thank you.